Today we are going to discuss differentiation of primary tissues in roots. Now firstly, what is primary growth? Okay. So whatever growth takes place during the initial stages is called primary growth, and the tissues formed during that period are called primary tissues. So how do we define it? Primary growth, it is the increase in length of shoots and roots in plants it is result of cell division in the apical meristems. Okay. So increase in length, the lengthwise growth, that is called primary growth. It takes place in shoots as well as in roots in plants, and it is because of the activity of apical meristem. Now what primary meristems are present in root apex? So the root apex has 3 primary meristems. They are protoderm, ground meristem, and procambium. In a previous lecture, we discussed that these are the terms proposed by Heberland. Okay. So look at the diagram. We have already studied this diagram when we discussed the structure of root apex. So this is a real cross section of root apex. It is a longitudinal section. This outermost region where the cells are slightly transparent, this is root cap area. Okay. And this portion that we showed in a parallel row of cells, it is calyptrogen. Then we have few initials, and from those initials there is formation of the central cylinder called procambium. Toward the outer side we have rectangular cells which are going to form protoderm, and in between zone is called ground meristem. So you can see here clearly that the cells in this region, this much portion is the root apex, means it is an inverted cup like structure where the actively dividing cells are present, and they are going to give rise to different histogens. So basically 3 primary meristems are present at the initial stages in a root apex: protoderm, ground meristem, and procambium. Then we observe an additional structure near the middle of apex. It is called quiescent center. It is an important term, quiescent center. What is quiescent center? In this area, cells divide slowly than other meristematic cells. These are relatively resistant to damage from radiation and toxic chemicals. So, these are resistant cells which are less dividing, and what is their function? Why they are present in the root apex? Their function is that they may act as reserve cells that can restore the meristem if it becomes damaged. Okay? Because roots, they are growing underground. So, there is abrasion, continuous abrasion because of the soil particles. So, it may happen that by chance the root apex gets damaged. So, these cells, they serve as the reserve cells. So, they are going to divide at that particular time if the apex is damaged. So, it will give rise to the meristematic apex. That is the function of quiescent center. So, now what happened to these primary meristems? These three primary meristems, they are going to give rise to primary tissue systems, that is three primary meristems give rise to three primary tissues systems. Now, what are those systems? That is dermal, vascular and ground tissue system. Okay. So, what will be the fate of these, that we will discuss later on. But how the root grows? We discussed earlier that the root apex has 4 different zones. At the tip there is root cap, then there is a zone of meristematic cells. As we move up, there is a zone of elongated cells, and afterwards when the cells, they get differentiating, that forms the zone of maturation. Okay. So, how this happens? What changes they take place? So, if we talk about the zone of root apex, in zones of root apex, the meristematic cells, means the second zone, add layer of new cells which move towards elongation zone where they elongate 10 times the original size. Okay. So, their length increases, that is why it is called elongation zone, and the size may reach approximately 10 times the original size of the cell produced by meristem. Then they move towards, now the next zone, fourth one, the zone of maturation. Where cells specialize, or we call it differentiate. In structure and function. Okay. So, they will reach the maturation zone, and now they are acquiring a particular shape, a structure, and the function is assigned accordingly. So, it means now from these 3 meristems, these three tissue systems are going to develop, that is in the zone of maturation. So, in this region all three tissue systems complete differentiation and become functional. Okay, the system that has to form the outermost layer, epidermis, they will start functioning accordingly. The ones that will be developing into the cortical region, the ground tissue, they will assume their functions, and similarly the ones which are present in the center, they will start behaving as the vascular tissues. It means they will start the conduction of water and food. So, accordingly in the maturation zone, they acquire different functions. So, three primary tissue systems they develop. They are dermal tissue, ground tissue, and vascular tissue. So, these are the primary tissues developed from the meristematic cells. Now, what will be their fate? They are going to mature into what layers of the root during primary growth. So, the dermal tissue will give rise to epidermis and root hair. Ground tissue is going to give rise to endodermis, cortex and pith, and vascular tissue is going to give rise to stele, pericycle, and stele includes xylem and phloem. Okay, so, these are the tissues which are going to develop finally in a root showing primary growth. So, what are the functions of these different layers? Epidermis, it provides protection and helps in absorption of water and minerals. Root hairs, they increase surface area of root and contribute to absorption. Okay, so, that is the fate of the dermal tissue. Now, ground tissue, epidermis, it serves as checkpoint for materials entering the vascular system through Casparian strips. What are Casparian strips? These are the thickenings of a waxy substance called suberin. okay, and they are present along the inner tangential wall and radial walls of the endodermal cells, so as to restrict the entry of material along cell walls. And finally, the material or the water, it has to enter the cytoplasm of endodermal cells, only then it can reach the vascular cylinder. So, they are present there in order to check the entry of different materials into vascular system. So, that is a function of endodermis. Now, second we have cortex. It is meant to store different substances okay. So, this is meant to store different substances, and same is a function of pith as well. These are simple parenchymatous cells, and the cells of the cortex, they especially they store starch grains, or in some cases we observe a differentiated layer of hypodermis as well from the outer cortical cells, and they may either be photosynthetic if we talk about hydrophytes, if they are photosynthetic, or they may simply store the materials, or they may become collenchymatous. Okay, now coming to the vascular tissue. The vascular tissue is going to differentiate into stele and pericycle. So, the pericycle give rise to lateral roots. We have discussed the structure of roots, where we mentioned lateral roots include secondary roots, tertiary roots and rootlets. So, they arise from pericycle. And stele, which develops into xylem and phloem. Its function is conduction of water if we are talking about xylem, and phloem if we are talking about and food if we are talking about phloem. But their arrangement varies. Okay. If it is a dicot root, then these elements are present in X shape, and if it is a monocot root, then the bundles are arranged in the form of a ring around pith. In dicot roots, generally the pith is absent. If present, very few cells are present in the center, whereas in monocot roots, a well developed pith is present in the center, around which the vascular bundles develop in the form of a ring. But otherwise, in root, the bundles are radial. Xylem alternates with phloem. So, if we look at the structure, see, this is the structure of a root. Okay. Here I am going to show you the layers that are developed from various tissue systems. So, we discussed the outermost layer is epidermis. This is epidermis, followed by the exodermis. Exodermis is present in certain monocots in which the outer layer of cortex becomes sclerenchymatous. It is not present in all the roots. Okay. Then these are the inner layers of cortex, which are parenchymatous, parenchyma cells. Then the green color that is shown here, that marks the innermost layer of cortex called endodermis, and here the thickenings have been shown along the inner tangential and radial walls, and those thickenings are called Casparian thickenings. Then we have another layer which is called pericycle. So, pericycle, the function is to give rise to lateral roots. That is why we say the lateral roots are endogenous in origin, means they are arising from pericycle. And in the center we have the stellar region, in which there is xylem and the alternate patches of phloem. So, always the arrangement is like X shape. So, we draw it like xylem in the center. Let us suppose this portion is xylem and phloem in the alternate patches. So, the bundles are called radial vascular bundles. Okay. This is a case of dicots. I am going to show you another diagram. See, this is a comparison of the dicot and monocot root. So, in dicot, the arrangement of xylem is in a X form, whereas if we see monocots, a well developed pith is present in the center and bundles are arranged in the form of a ring. Okay. Then they are showing, this is just a representation to show pericycle, endodermis, cortex, epidermis. So, these are the various layers which develop from primary meristems. These are the mature cells, differentiated cells which are having their own particular function as well as structure. Okay. So, that was related to the structure. So, this is the finally the primary root shows growth lengthwise and grows towards the deeper layers of soil due to activity of meristems okay. So, that is the main purpose of primary growth, that the length of root increases so that it can enter the deeper layers of soil in order to absorb water. So, the main function is water absorption. Okay. And if generally we see the proportion of root and shoot in a plant, it is almost 50-50. So, whatever growth you see upside means above ground, the root part, and the growth below ground, the root part. So, the shoot and root ratio is almost 1:1. Okay. So, that was all about the primary growth of root.